Welcome to the CDC. We are here today with representatives from Pfizer to discuss their newest mRNA vaccine, the SARS-CoV-2 vaccine. We will walk you through what mRNA is, what an mRNA vaccine is, and how the Pfizer COVID-19 vaccine works and is produced on a large scale. Our main goal is to educate the public about the COVID-19 vaccine in order to encourage everyone to get vaccinated. Let's start with some overview on what mRNA is and how an mRNA vaccine works. In our own bodies, mRNA transcription and translation is occurring all of the time. Essentially, the purpose of transcription and translation is to create a protein. The code for this protein is within a specific gene in our DNA. Transcription begins in our own bodies by an enzyme, RNA polymerase, unzipping our DNA from its double helix. This allows for complementary base pairing to occur based off of the region of interest in our DNA. These complementary base pairs are joined together to create single-stranded mRNA. Once the mRNA strand is complete, it detaches from our DNA. This newly created mRNA then moves to the ribosome of the cell. Once there, the process of translation, or creating a protein from the mRNA, is started. Every mRNA strand is made up of codons, which are triplet codes made of 3 nucleotides or bases. These codons all represent specific amino acids. The ribosome is able to read these codons and translate them into ordered strings of amino acids which form proteins. In an mRNA vaccine, the transcription step is done outside of our bodies. The single-stranded mRNA is created in the lab from the DNA of the coronavirus cells. This pre-made mRNA is then injected into our bodies when we receive the vaccine. Once inside of us, it is able to make its way to our cells and begin the translation process, where the injected mRNA is translated into a specific protein. But what protein from SARS-CoV-2 is being injected inside of us when we get the vaccine? One thing to remember is that the mRNA vaccine does not give you a weakened or live form of the coronavirus, unlike typical antibody vaccines. Instead, the mRNA in the vaccine is encoded to create the surface glycoprotein of COVID, also known as the spike protein. This is the protein that gives the coronavirus cells their crown-like shape, as the spike protein sits directly on the cell's surface. Once the mRNA for the spike protein is in our cells, we begin making this protein. As soon as translation of the mRNA is completed, the mRNA encoding the instructions for this protein is immediately broken down and destroyed. The spike protein that was formed, however, survives and moves to the surface of our own cells. Our immune system will immediately recognize these spike proteins as foreign, creating antibodies to destroy the cells with the spike proteins on their surface. Thus, when a true SARS-CoV-2 virus cell enters your body with a spike protein on it, our immune system will already know how to form antibodies against this virus in order to destroy it.
The mRNA construct used in the Pfizer vaccine is a nucleoside-modified messenger RNA, also known as modRNA. This messenger RNA construct encodes for the full spike glycoprotein that COVID-19 contains. The spike glycoprotein protrudes from the outside of mature coronavirus particles, which makes it a good target for immune responses and neutralizing antibodies. The spike protein contains 2 subunits, S1 and S2, which the mRNA construct both encodes for. S1 is the subunit that contains the N-terminal domain and the receptor binding domain. S2 is the subunit that contains the fusion protein, connecting region, heptide repeat 1, heptide repeat 2, and central helix. This spike protein in COVID has 2 main states, a prefusion state and a postfusion state, wherein the spike changes its conformation. Normally, this postfusion state occurs after the virus has fused with the cell membrane. Pfizer wants to elicit an immune response for the prefusion state of the spike protein, which isn't exactly very stable when encoded. In order to remedy this, the encoding messenger RNA coding for the spike protein contains a mutation that adds 2 prolines in place of the K986 and V987 residues to the encoded spike protein, which helps in maintaining that prefusion stability. However, as we know, mRNA is very unstable, and for it to reach its desired place within the cell for encoding, scientists package it into liquid nanoparticle. This prevents the degradation of the mRNA and enables it to enter the cell.
mRNA vaccines use a spike protein which is found on the surface of a target virus, containing instructions for cells. Since mRNA cannot be cloned directly, synthetic mRNA can be created on the basis of spike proteins by using a DNA copy of the original RNA. This can be done in vitro by utilizing a copy DNA template that is transcribed using RNA polymerase. After this vaccine is given intravenously, the mRNA instructions go into the immune cells, and then tell them how to make the protein piece. While traditional vaccines stimulate an antibody response by directly injecting antigens, a weakened virus, or a carrier virus engineered to have antigens, mRNA vaccines instead directly inject a synthetic RNA sequence and let the body's internal machinery do the work. The key factor behind determining how to encode the immunogen is the fact that the spike protein, which are the characteristic protruding surface antigens of coronaviruses, are what help the virus attach and inject DNA into host cells. Antibodies in our immune system will then bind to these spikes and prevent them from attaching into our cells. All or part of the spike protein is injected as a part of the nucleoside modified mRNA. The code is then broken down by proteasomes. Then, class 1 and class 2 MHC molecules attach to the encoded antigen and transport it into the cellular membrane, thereby activating the dendritic cell. While non-immune cells can potentially absorb this mRNA, dendritic cells do it much more quickly. Once the dendritic cells are activated, they migrate to the lymph nodes where the antigen is presented to the T cells and the B cells. The dendritic cells display the spike protein on the surface and then pose as an antigen, allowing the T and B cells to create the necessary antibodies that can specifically bind to the coronavirus surface spikes, preparing us in case we are exposed to the actual virus. The instructions are then broken down and destroyed.
With the vaccine approved for distribution, it becomes necessary to produce this product in bulk. However, the manufacturing process for vaccines is typically developed over the course of many years due to technical, quality, and financial constraints. Due to the immediate need and scale of the coronavirus pandemic, the manufacturing process has been greatly accelerated. Pfizer engineers successfully created and scaled a process in under a year by ordering materials and developing processes for potential vaccine candidates ahead of the official approval, and integrated important quality guidelines and checks implemented at various checkpoints. Rather than concentrating the process at one location, which takes additional time and resources, multiple locations were modified and adapted based on sustainability or suitability for different stages of the vaccine production process. The current process involves 25 different suppliers across 19 countries. The exact detailed process is So, generally speaking, however, the process for manufacturing these vaccines comes in 3 steps divided among 3 locations. First, the DNA plasmids used to create a vaccine template are generated in a factory located in St. Louis, Missouri. Creating these critical raw materials takes 2 weeks, not including the quality assurance process that follows. Next, the plasmids are transported to Andover, Massachusetts, where the plasmids are translated into mRNA in a 4-day batch process. Finally, the mRNA materials are formulated, placed inside the lipid nanoparticles, and filled into vials at a facility in Kalamazoo, Michigan. This location will be expanded to include a new formulation site within the next year. The process here takes over 1 week to generate the lipid coatings, package, and freeze. At each step of the process, quality assurance checks are conducted to ensure no tampering, degradation, or errors have occurred to jeopardize the safety and efficacy of the vaccines. This final checkpoint consists of a 2 to 3 week assessment comprising over 40 quality assurance tests. From there, the vials must be transported and stored in a cold chain network at -80 degrees Celsius. There have been several limiting factors in the manufacturing and rollout of the Pfizer vaccine. The 2 main factors are time and temperature. Since the Pfizer vaccine was first authorized for emergency use by the Food and Drugs Administration, or FDA, in December of 2020, over 29 million doses have been administered as of February. Pfizer produces over 5 million doses per week. The process initially took 110 days, which was severely limiting for a 2-dose product. However, Pfizer is working on decreasing this time by 60 days by increasing its production yield and streamlining the quality testing timeline.
The purification step is just as important as the synthesis itself. RNA transcription does not necessarily yield the mRNA that we want in the vaccine. The mixture after the synthesis potentially contains double-stranded RNA, host DNA, RNA, proteins, and other molecules that were used to stabilize enzymes. These molecules are usually very large and can aggregate, and they will trigger an inflammatory response before the mRNA has had the chance to enter the cells and translate to produce the spike proteins. Several methods are used to purify mRNA. The very first step is to ultracentrifuge the solution and filter out the large molecules. The following chromatography methods use the differences in affinity to the column and the corresponding elution times. Hybridization affinity chromatography is a sequence-specific purification method that utilizes oligopolythymidine ligand, which captures mRNA by its poly-A tail, which is a chain of adenine nucleotides, and bonds to it by the AT base pairing. Next, the hydrophobic interaction chromatography uses the hydrophobic interactions between the column and mRNA of interest. This method is used to filter out single-stranded DNA from double-strand DNA and other incomplete transcriptions. And lastly, anion exchange chromatography can also eliminate double-strand RNA and DNA by the affinity of the charged molecules to the ion exchanger column. Each chromatography method was washed with proper buffer or salt over the time to change the affinity of mRNA molecules to the column. These purification methods are later verified using different methods such as high-performance liquid chromatography, SCS-PAGE, and dynamic light scattering.
Before the Pfizer COVID-19 vaccine was approved for emergency use by the FDA, numerous clinical trials had to be run to test the efficacy and side effects of the mRNA vaccine. The vaccine is currently contradicted for people with severe allergies. The Phase 3 clinical trial results were released to the public December 10, 2020. In this trial, 43,448 people, all 16 years or older, participated in the trial. Half of these first participants were given a placebo, and the other half the real mRNA vaccine. The results from our clinical trial indicate that our vaccine is highly effective at preventing the COVID-19 virus. The vaccine was determined to be 95% effective, starting about 7 days after the second dose. This data was determined from the fact that only 8 participants in the vaccine group tested positive for COVID, while 162 did in the placebo group. Not only this, but the results of the trial also showed the vaccine has a strong safety profile. The most common side effects were fatigue, fever, or pain in the injection area, all of which went away in 2 days or less, and severe reactions to the vaccine only occurred in less than 2% of participants. It is important to keep in mind, however, that the vaccine will not prevent you from getting infected by the virus. However, it will reduce the severity of the symptoms. Even if you were administered the vaccine, it is important to keep your mask on and social distance for those who have not been vaccinated yet. Reach out to your local health officials to see when you are eligible for the vaccine. More information can be found on cdc.gov.